like a drum head would be. And so when that sound wave travels down the canal and hits the eardrum, it makes the eardrum start vibrating. And these auditory ossicles here, you can see how it touches the eardrum. And so these auditory ossicles start vibrating. It really concentrates these little tiny bones here. By really kind of narrowing down the sound wave, it really helps concentrate the sound and the vibration so that we can actually distinguish what it is. So these ossicles then transfer that vibration here to the inner ear. And the inner ear is actually, this is all bone. Okay, so it's like a snail shell bone. These guys are all bones, but those are important for balance. And so those bones, though, are filled with fluid. And so the vibration that's coming in here off of this, off of the stapes, off of that last auditory ossicle, is going to trigger the fluid in the cochlea. That's the snail shell. It's going to trigger the fluid in the cochlea to begin to vibrate. And so now you have this fluid that is making waves. Okay? And so if the fluid makes waves, and I've got these little hair cells, they're going to start to bend. And so now I get my mechanical or physical change, okay? And then that information is sent along the vestibulocochlear nerve back to the brain, back to the temporal lobe of the brain to be processed. Okay? So we've got one more video on that, and then after that video, we are all set with the nervous system.
The ear is responsible for translating variations in air pressure, whether from speech, music, or other sources, into the neural activity necessary for our perception and interpretation of sound. The ear can be divided into three principal sections: the outer ear, the middle ear, and the inner ear. Each of these parts performs a specific function in processing sound information. Sound waves are first collected by the outer ear, which consists of the external ear, or pinna, and a canal that leads to the eardrum. The configuration of the external ear amplifies sound, particularly at the frequency ranges of 2,000 to 5,000 hertz. Not coincidentally, most human speech sounds are distributed in the frequency range around 3,000 hertz. From the ear canal, the sound waves vibrate the eardrum, which in turn vibrates three tiny bones in the middle ear: the malleus, incus, and stapes. The stapes vibrates a small membrane at the base of the cochlea, the oval window, which transmits amplified vibrational energy to the fluids of the cochlea. The round window serves to dampen the movement of fluid within the cochlea. The complex structures of the inner ear convert sound into neural activity. In mammals, the auditory portion of the inner ear is a coiled structure called the cochlea. The region nearest the oval window membrane is the base of the spiral. The other end, or top, is referred to as the apex. Along the length of the cochlea are three parallel canals: the tympanic canal, the vestibular canal, and the middle canal. The principal elements for converting sounds into neural activity are found on the basilar membrane, a flexible structure that separates the tympanic canal from the middle canal. Let's take a closer look at the basilar membrane by unrolling the cochlea and peering inside. The basilar membrane is about 5 times wider at the apex of the cochlea than at the base, even though the cochlea itself narrows toward its apex. It vibrates in response to sound transmitted to the fluid-filled cochlea by deflections of the oval window initiated by the bones of the middle ear. High-frequency sounds displace the narrow, stiff base of the basilar membrane more than they displace the wider, more flexible apex. Mid-frequency sound maximally displace the middle of the basilar membrane. Finally, lower frequency sounds maximally displace the apex. Within the middle canal and atop the basilar membrane is the organ of Corti, the collective term for all the elements involved in the transduction of sounds. The organ of Corti includes 3 main structures: the sensory cells, or hair cells, an elaborate framework of supporting cells, and the terminations of the auditory nerve fibers. The heights of the stereocilia increase progressively across the hair cell, so the tops approximate an inclined plane. Atop the organ of Corti is the tectorial membrane. The stereocilia of the outer hair cells extend into the indentations in the bottom of the tectorial membrane. The movements of fluid in the cochlea produce vibrations of the basilar membrane. These vibrations bend the stereocilia inserted into the tectorial membrane. Depending on the direction of the bend, the hair cells will either increase or decrease the firing rate of auditory nerve fibers.